are the single-celled organisms. So amoeba, paramecium, and bacteria, Escherichia coli, bacteria, E. coli, Escherichia coli, etc. So protozoan organisms, amoeba, paramecium, pigrina, and bacteria, Escherichia coli are the unicellular organisms or single-celled organisms. And next, multicellular organisms examples, you know already, what is fungi and plants and animals, multicellular organisms examples. So they are the single cellular organisms, multicellular organisms, unicellular organisms and multicellular organisms. So what are the differences of unicellular and multicellular organisms? Unicellular organisms made up of only one cell or single cell, and multicellular organisms are made up of more than one type of cell. A single cell performs single cell organisms, there no single cell organisms. A single cell performs all the life processes like obtaining food, respiration, excretion, growth and reproduction. Single cell. Next, multicellular organisms, there know all the activities, respiration, excretion, reproduction, and all the activities are carried by a variety of cells. So here a single cell, unicell, the unicell is performing all the activities. One and only cell is performing all the activities. But here in multicellular organisms, they performed number of cells there, no? So the number of cells are performing number of activities, all the activities. Examples: fungi, plants and animals. Okay, now clear? And next, so I would like to explain you some different types of cells. So here I would like to explain about observing a matchstick. So I wanted to observe cells in the matchstick. So observing a matchstick cells. Observing matchstick cells. Observing matchstick cells. So for this there is a process how we will observe this matchstick. So already you learned how the slide is prepared. So the slide will be prepared. So a transparent glass slide means a transparent glass. So that is microscopic slide is prepared on a 2 mm thick. So I am telling you slide preparation. Slide preparation. So we need to prepare slide to observe under the microscope. So before going to observe under the microscope, we need to make the slide. That's why I am telling the process of make the slide. So microscopic slide is prepared on 2 mm thick, 3 centimeters to 8 centimeters, 3 millimeters thick, and 3 centimeters into 8 centimeters. 3 centimeters and 8 centimeters rectangular strip. Rectangular strip. Clear? Huh? So and clean glass piece. Transparent glass piece. Transparent glass piece And next, if the object is thin and flat, can be directly placed on the glass, slide towards the in the drop of water with the help of soft and fine brush. A drop of glycerin is added to making the slide, and we will put the one drop of water on this slide. Why we are adding glycerin to the slide? Because of the object is kept for a longer time. Glycerin saves the material from drying, dehydration. So if the object is thick and cut it into the thin, for example, I wanted to observe mastic, then if I kept the mastic under the microscope, this mastic won't visible. So how we can see, how we can observe under the microscope? First of all, we will take a thin slice, a thin portion from that object, nearly 0.5 mm thick or less. So thick section with the help of sharp razor. So if you wanted to observe any of the object, you want to take a very small 0.5 mm thickness of material, we will collect and we will place on the slide, and then you can observe under the microscope. So a transparent glass, take a transparent glass the size of 2 millimeters of thick and 3 centimeters cross, 8 centimeters rectangular strip, and then the material in a drop of water and cover with a cover slip. The cover slip is one of the transparent glass, that is 0.1 mm cover slip. 0.5 object and 0.1 mm object, 0.5 mm cover slip. So cover slip, after preparation of slide, we have to cover the object with cover slip. So because we will store this is long, so many days. For example, the material in the drop of water and cover with cover slip. Do it while it is doing, do it carefully. And next, hold the cover slip over the object without forming air bubbles. So while you are placing the cover slip on this slide, so you have to take care while placing the cover slip, while covering the object with cover slip, without allowing air bubbles. If the air bubbles are allowed in that slide, so the image won't visible clearly. So that's why you have to take care. And extra water, if any, coming out from the edges of the cover slips may be dried. A piece of clothing or filter paper, you have to take it away from that slide. So this is the slide preparation. Have you clear, children? So the slide is ready. So then you can observe under the microscope that particular slide, that image will be visible to us very clearly. Okay. And next, yes, I will explain about observing mastic cells. Observing mastic cells. Okay, now observing mastic cells. Take a mastic and soak it for half an hour. That is the first point. Take a mastic and soak it for half an hour. Take a mastic and soak it for half an hour. and cut thin slices of it. Cut thin slices of it and cut thin slices of it. Slices of it. So what is the first point? Take a matchstick and soak it for half an hour and cut thin slices of it. And select a thin slice and place it on a slide. Select a thin slice and place it on a slide. So next, okay. So select a thin slice and place it on a slide without allowing air bubbles and observe it under the microscope. So if you observe under the microscope, the picture is visible to us like this. So a rectangular shaped like structures, visible to us. So those rectangular, each and every rectangular shape of empty space is the cell. That is the cell. These type of cells are observed by Robert Hooke. Okay, clear? Huh? So this is the mastic cells. What is? So I observed mastic cells under the microscope. Under the microscope, the structures visible like this. Okay, clear? Huh? Okay. Next, I will continue tomorrow. Okay, one more point. Actually, mastic cells are the dead cells. These are the dead cells. Actually, the discovery of cell by Robert Hooke was a milestone in the history of science. So you know already. Yes, the book. There is a book, the History of Science book. There, no. In the previous lesson we learned the History of Science book written by F. Kajori. History of Science book written by F. Kajori. So in the history of science, the cell discovery is the milestone to discovery by Robert Hooke. So these are the dead cells. Actually, these are the dead cells. So like this way, the cells of cork and of mastic, dead cells. So can we see living cells under the microscope? We can see. Okay, we will learn coming lessons.